Well, anyway, it's an enormous pleasure to welcome you all on this quite gorgeous fall day to our third annual Population Health Sciences Symposium. I'm expecting that bit by bit we will, the crowd will flesh out, but wanted to just dive right in. You know, most of our sister centers and institutes take advantage of their annual symposium as a way of showcasing the fabulous achievements that they've had in the past. And indeed, in our previous symposium, we tried to highlight on purpose things that were in motion and had produced some results. Today, we're going to try something a bit different, and it's a little bit of an experiment, which is that rather than in largely looking backwards at things that we have achieved, although we will talk about some of those things in the course of the day, I hope, we want this to be extremely forward-looking, and our goal today is to achieve what we've tried to epitomize here on our little T-shirt, which is connect. We're trying to show how the various kinds of data that we've collected, and you can see where, as you'll see, we're collecting various kinds of partners and data from around the world to see what the opportunities may be in bringing together folks from all 7 of the colleges here at Stanford, all 7 of the schools, to meaningfully do work on various kinds of data and various populations that might not, within the confines of the agendas of the individual schools, seem obvious or even necessarily all that relevant. And so what we've done today is pick 5 broad collaborations that we have in motion as we speak today in one form or another, and try and provocatively present a little bit about what the data and the population are about. And I'm going to start in a couple of moments talking about the first of the 5. And then have someone speak a little bit about both what can be done or has been done or some combination thereof before we have a brief panel discussion to talk about where the opportunities may be. And again, the focus on the day is on those unique opportunities. In any event, before we start, a couple of housekeeping things. I hope everyone figured out a way, one way or another, to park. We do have free parking up at Galvez. And if anyone needs to know the magic number to put in so that it's really free, grab any of my colleagues and staff, most of whom, unlike me, are wearing these gorgeous blue T-shirts, so they should be pretty easy to find. Needless to say, good time to turn off your cell phone. Mine decided to do it on its own this morning. I'm not quite sure why, but greeted me with a morning shutdown. But if yours didn't, this would be a great time to turn it off. For those of you who aren't familiar with Arriaga, the restrooms are, there's one set right here around the corner. I think most of you found the coffee as the morning warms up. It'll maybe be more attractive than it's been. There's some breakfast. And again, many of my colleagues and staff are here and should be quite readily identifiable by the blue T-shirts. In any event, with that, I want to now move on to talk about our very first data opportunity or partnership opportunity, and that is with the health center in Israel known as Clalit. So for those of you who don't know it, Israel is one of the countries in the world that's been now for well over 20 years conducting a rather interesting healthcare experiment. They have a single payer system, and all Israelis have the opportunity at birth of declaring themselves a member of one of 4 highly competitive healthcare systems. The healthcare systems largely are reimbursed by a single fee per person, so it's completely capitated in a Kaiser-like fashion. And we were lucky enough through relationship that Laron Einhoff in the econ department had with Ron Belliser, who's the director of their research institute at Clalit, the largest of the these 4 healthcare systems in Israel. To be able to forge a relationship. We've gotten some wonderful help from development to be able to fuel it with some early funds, hire some faculty and staff on the Tel Aviv side of this relationship, and to start moving forward. So I just want to spend a minute or two telling you about what the data opportunity looks like. The healthcare system itself has a century of history, but only in the past 20 years has virtually everything that they do been digitized, which of course makes it relevant to us. It's far and away the largest of the 4 healthcare systems, having now just modestly over 50% of the entire population. And for historic reasons, they provide care to most of the underserved population of the country. Again, remember, they're not competing on price here, they're competing on service, which is the basis for the success or failure of the organizations. As you can see, they have an enormous network of clinics as well as hospitals, and provide all of pretty much complete healthcare services from primary care all the way out to quaternary. In fact, for those who are interested in the health services side of things, it's a very primary care driven system in which most of the competition over these systems is at the primary care level. People view the hospitals as competing with themselves in a separate kind of a way. This is some just general account of the population. It's about almost 4.5 million current members. They have active records, or they have records on 6 million, since again it's population based, and you can see what the ethnic composition is. Even within the Jewish population, an incredible diversity. The world's largest population of Ashkenazi Jews, which, as you know, is a genetically quite interesting population, a large portion of Sephardic Jews, and then a wide variety of minority and smaller groups like the Ethiopian Jews and so forth. As you can see, the nature of the population is such that it's got a very, very high rate of various genetic mutations, and obviously unusual and interesting cultural and environmental diversity across that population. And finally, this is what the data trove looks like. So you can imagine it's a claims data set, sort of, with electronic medical records thrown in, some linkages to population level social and demographic data, geographic GPS coded population. We imagined, of course, when we started with this, that this would be a great claim set, and people interested in comparative healthcare systems and so forth would start playing with it. And indeed, that is where we started. Laurent himself, working with Ron, did a lovely study looking at readmission after congestive heart failure, followed it with a study now mid-course looking at end-of-life care, in particular the cost of care during the last year of life, with a focus on cancer care. Very traditional health services research. Julie Parsonet, who couldn't be with us, she's in Oxford today, had the cool idea of, because of the generational aspect, of looking at some generational aspects of the relationship between the microbiome and the onset of asthma early in life, in particular the relationship between antibiotic use and that. But it wasn't until I actually, almost by accident, mentioned this new opportunity to Manny Rivas, who will talk to you a little bit about his thoughts, that I began to realize what the potential in this data set might actually be. So Manny is a computational geneticist who some of you have hopefully had the pleasure to meet. He's on a meteoric rise in terms of the scope of his work and the attention that it's getting. But Manny just filled us with a wide repertoire of potential study opportunities in his space. And as we'll learn from the panel, I think there are even yet more opportunities still moving far away from traditional health services related research. So with that, I will hand the microphone off to Dr. Rivas and get us started.
All right, thank you, Mark. First, I'd like to thank PHS for inviting me to share with you a vision that we have at the Department of Biomedical Data Science in terms of opportunities that exist with combining medical population data with human genetic data and new data science technologies that may be developed by researchers at Stanford. So there's a phenomenal opportunity in Clalit to come up with groundbreaking discoveries. And what I'd like to do in this presentation is to focus on two different stories that hopefully by the end will highlight the potential that exists in this phenomenal system. So I'll briefly go over the slides the first few slides, I think Mark basically has introduced Clalit. So what I'll just reiterate is that Clalit makes up about 54% of members in Israel are members of Clalit Healthcare system. There are over 1,500 clinics in Clalit and owns and operates 14 hospitals, fantastic network of pharmacies, imaging centers, and labs. And there's comprehensive clinical and administrative data that's been collected since 2000. So you can imagine the phenomenal phenotyping data set that's available on over 4 million individuals that are part of this healthcare system. One of the points that Mark alluded to is that in Israel, there's a phenomenal population diversity, and it has wide-range implications for research. For example, in the context of the population composition of Israel, it's composed of about 75% Jews of different ethnic origins, 20% Arabs, and 4.2% of other population groups. And these population makeup ends up influencing the predisposition to or risks to diseases. And some diseases are observed at different prevalences in these population groups. There's a phenomenal opportunity to layer in on top of these data sets human genetic data. So what I'd like to do is to give you just a highlight as to an example as to how human genetic data can improve your interpretation of human diseases. Here's an example of a founder effect allele, where there's an observation of a frame shift indel in this gene called HEX-A. In European populations, or non-Finnish European populations, the frequency of this particular allele is about 1 in 1,000. That means that one individual in about 1,000 individuals carries this allele. In the Ashkenazi Jewish population, however, we see this allele in 1 in 100 individuals. It's a typical example of a founder effect allele, and it has implication for the diseases that are observed in some of these populations. This particular allele has a relationship to Tay-Sachs disease. So just to give you a context in terms of other projects as to what opportunities exist, I'll give you a preview of human genetic data that's been generated in one of the populations that's relevant to Clalit. In a study conducted a few years ago, we generated exome sequencing data in approximately 6,000 Ashkenazi Jewish individuals, half of which are individuals that have inflammatory bowel disease, which is comprised of Crohn's disease and ulcerative colitis. When you take that human genetic data and you try to identify alleles that seem to be enriched in these particular populations, you start seeing overlaps with previously annotated disease predisposing alleles, which is quite relevant in understanding how risk to diseases in certain populations is attributed to human genetics. For example, in the context of Ashkenazi Jewish population, we were able to identify 151 alleles in ClinVar that are labeled as pathogenic. This is analogous to what you see in other populations, for example, in Finland, where this similar experiment has been done, where we've been able to perform exome sequencing and get an aggregation of allele frequencies across that population. Now, one complex trait that I did want to focus on is Crohn's disease, the Ashkenazi Jewish population. Crohn's disease has been documented with about a 2 to 4-fold increase in prevalence in Ashkenazi Jews versus non-Ashkenazi Jewish population. And it's unclear whether these differences in genetic risk exist and how they may contribute to differences in prevalence. So in this study, we were able to amass or combine genetic data along with the phenotype data for inflammatory bowel disease and identify genetic risk factors that seem to be enriched in the Ashkenazi Jewish population, and it gives us a better understanding as to how human genetic variants contribute to that disease predisposition. Now, another opportunity that exists is the ability to look at genome-wide genetic data. Now we're able to, I've given you examples as to how we looked at specific genetic variants, but now we've also been able to look at genome-wide genetic variant data and identify that there's a strong shift in polygenic genetic risk in the Ashkenazi Jewish population versus non-Ashkenazi Jewish population, even when you exclude these alleles in the previously implicated Crohn's disease genes, NOD2 and LARG2. So here in this plot, for example, you can see that the genetic risk scores for Ashkenazi Jewish individuals shown by the green density, are shifted to the right compared to non-Ashkenazi Jewish individuals. The fantastic opportunity that exists is being able to decompose how those differences in genetic risk are attributed to specific genes or genetic variants. And what we've done in this study is to characterize where is it that in the genome where this increased shift in genetic risk is coming from. The cool thing about this particular study is that we were able to pinpoint that it is, in fact, sets of alleles that contribute to bacterial response that gives rise to this increase in disease risk. Now, just to finish off, one of the things that I do want to highlight is the opportunities that exist when you combine population medical data with human genetic data to empower new data science technologies. One population cohort that thankfully PHS has brought into Stanford is the UK Biobank data. It enables us to study the shared genetics with other diseases and phenotypes. The UK Biobank is comprised of about 500,000 participants. And in my lab, what we've done is to create resources that other researchers around the world can use to query some of that human genetic association data. This was set up in the first year of the lab with the students in the lab, along with some phenomenal rotation students. And you're more than welcome to browse some of the association data as of right now. So far, it's had a very positive reception with over 1,500 users and over 3,500 sessions. Those sessions have about 8 to 10 minutes, meaning that people are actually spending a good amount of time in the Global Biobank engine. And one of the things that I do want to highlight is the opportunity that exists when you do have human genetic data combined with this disease outcome data sets in order to identify genetic variants that may lead to insights into therapeutic hypotheses. And I think that's a fantastic opportunity that exists with the Clalit data set that would be worthwhile to move forward with. One opportunity that exists with rare effect alleles, especially enriched alleles in particular populations, is the ability to identify genetic mutations that protect against human diseases instead of giving rise to human disease. Pharmaceutical companies are quite excited about these opportunities, and these are two examples that we've identified in the context of Crohn's disease. The specific example of CARD9 at the top, where we identified a mutation that confers about a 4-fold protection to Crohn's disease, and the example of RNF186, where we identified a mutation that confers about a 2-fold protection to ulcerative colitis. This is in contrast to what's observed in most human genetic studies with common variants, where you see pretty small effects near zero. Here now, as we move to rare variants, we're starting to see very strong effects, which is a fantastic opportunity to understand human disease biology. So in the context of having access to population medical data, what we did with the UK Biobank was to use that data and those genetic variants that are predicted to have strong effects on gene function to start to identify alleles that may have strong effects on disease risk. So we combined medical record data across 300,000 individuals in the UK Biobank, along with the human genetic data, and started trying to identify these strong effect genetic variants. Just to give you some context in the types of discoveries that you can come up with, here in this bottom left plot, what you can see is a type of phenomenon that's observed nowadays with human genetic data, that with common variants, most of the effects are centered near zero, and this phenomenon of increased effects as you go to rare frequencies in the spectrum of, the psi frequency spectrum of human genetic variation, you're starting to identify alleles that have pretty strong effects. One of the findings that I do want to highlight is the identification of a protective allele in IL-33, which is at the bottom left. Now, that genetic variant confers over a 2-fold protective effect on asthma disease risk. The great thing about that discovery is that it gives you a new signal for therapeutic hypotheses for moving forward with some therapeutic drug development pipeline. So, all in all, this study is summarized in a bioRxiv paper, and I just want to conclude by saying that there are ample opportunities in linking medical population data with human genetic data and new data science technologies. There is a great opportunity for Clalit to be one of the leading partners in our global search for understanding, treating, and preventing disease. And right now, with Mark and other folks at Stanford, we're proposing a pilot project in Clalit that focuses on Crohn's disease and ulcerative colitis, where the plan is to generate human genetic data in some of the patients in Clalit. So with that, I'd just like to thank folks in my lab and also acknowledge the contribution from folks in the IBD exome sequencing study, and highlight the contribution from Ron Ballister, Mark Cohen, and Heba, who have led the pilot project with Clalit. Thank you.
Well, thank you, Manny. What I'd like to do now is just briefly introduce our panel, many of whom I think you know. Sitting right here, Jeremy Fries, who is a relatively new professor of sociology in the institution, who has made a fabulous career of longitudinal studies of populations looking at the intersection between their social environment, their personal behavior and environment, and their biology. Sitting next to Jeremy is Carlos Bustamante. I think needs no big introduction in this crowd. Carlos is a very widely known computational geneticist and chair now of our new Department of Biomedical Data Science. Sitting next to him, Sandy Winter, who's a research scientist who in the Stanford Prevention Research Center, who has taken the lead in a global project now, the WELL project, which hopefully she'll tell us a little bit about, but perhaps more importantly, has been at the edge of exploring a variety of tools that can be used in populations for linking their social and physical environment with their health. So with no further ado, what I'd like to do is kick off the questions. I'm actually going to start, I think, with Sandy and ask you to tell us a little bit about some of those tools, the Discover tool in particular, and tell us how we might take advantage of the fact that unlike other populations like the UK Biobank, this is a living population in which we have access to the 4.4 million people in their clinical environment. And part of the excitement of that relationship is that we could actually use both on the clinical side, but also in other ways for obtaining information, full access to the population. Sandy?
Thank you very much, Mark. Can everybody hear me? Okay. So very differently to the work that Manny was describing, the work that our lab does, which is the Healthy Aging Research and Technology Solutions Lab, which is run by Dr. Abby King, we look at people's environments because we know that the environment in which people live impacts their ability to make and maintain healthy decisions. So the data that gets gathered in the kind of work that we do is not gathered in medical appointments, it's not genetic cellular level data. What we do is we engage with community residents. We've developed an app that can be done on smartphones or on tablets, and it allows community residents to go around their neighborhoods to document, using photographs and audio narratives, features of their environment that impact their ability to make healthy choices. So these could be things like the ability to engage in physical activity, which might be things to do with sidewalks or park availability. It could also be something to do with access to healthy foods. So some of the work that we've done in North San Mateo County among older adults, we found that sometimes older adults have to take sort of 2 buses and a train to get to the store that sells the food that they can afford to eat that's relevant to their cultures. We have some other groups who are working with older gay populations in Canada, looking at how people's environments helps them to feel safe or not safe. So the data that we gather is very different to the Clalit kind of data. It's gathered by community residents. Once the community residents gather the data, they then go through a collective process where they review the data and they prioritize issues to address. So the whole part of this project came about because when we were speaking to the county health departments and asking them about what kind of data they wanted, they said they have lots of data. The problem is that they need to hear the human stories behind how the data can be used most effectively. And so what our approach does, it allows this very personal telling of the features of people's environment that help or hinder their healthy choices. So we engage in a collective review of the data. The citizen scientists or community residents brainstorm solutions. So it's not all about problem finding, it's also a very solution-oriented approach. They think of solutions and they think of potential partners who they can work with to try and make improvements in the built environment. We've done this work in Israel. They were one of our most successful partners, which is why I think I get to sit here today. And we worked with all sorts of different groups, so not non-profit groups. We worked with Eshel, which is a large private-public partnership there. And the older adults that we were working with were able to meet with the mayor of Haifa, and the mayor of Haifa joined with this project very in full force, and they were able to do things like they mapped out a walking route where older adults can go for a walk that has things like the bathrooms that they need and the benches and things like that they need. And it was also very useful in bringing together people of different cultures. So we had Jewish Israelis and Arab Israelis. And the thing about the built environment is that it affects everybody equally, and it doesn't matter, what cultural background you come from. And so it was a melting pot where we were able to bring together people from these different cultures. It's also very intergenerational. We've done this project many times with older adults and with teenagers in North Fair Oaks. And it's interesting to see how older adults and youth view their environments differentially. And so I think in terms of matching it with the Clalit data, I think that it's important to think not only of the kinds of data that are gathered in medical institutions, but also the things like your built and social environment, which can have as much of an impact on your health. And so making these connections in a very solution-oriented way, I think, is an important way to move forward. And in Israel, the pilot project that we did that was in 3 cities with about 150 people is now being scaled up nationwide, and they're going to be gathering these kinds of data across the nation and engaging, as I said, in this very solution-oriented approach to trying to improve people's built environments.
So I'm going to push you a step further. Because one of the, at least from my point of view, most exciting opportunities, and again, I draw the distinction in with this particular population compared to many about which we have either information or biomaterial and the like, is that, of course, we actually have the people in the midst of their longitudinal trajectory through life. Could you imagine taking advantage of that, for example, as an evaluation tool to really assess whether or not, I mean, there's no question that some of this community discovery and intervention has some community benefit, which might be quite separate from demonstrable health benefit. But could we imagine using the fact that half of that very population on which you're scaling up now is under long-term observation, and therefore we don't have to recollect baseline information on their health status? We actually have baseline information, and assuming that the interventions are staged in a somewhat quasi-experimental way that would allow you to analyze them. Could we use this health information to evaluate what we're doing, and is it worth actually trying to get the outward-facing leadership of Clalit to think about this experiment that in some sense you've already launched, knowingly or otherwise?
Absolutely. So I think one of the problems for us as researchers when we get funded to do this kind of work is that it's on a very limited timescale. It's a 2 or 3 year project. And the problem with a lot of these changes that involve built and social environments is that they can take a long time. If you're going to put a new public transport system or a new food market in a desert, it can take years to see the effects of those changes. So I think being able to partner with data where there's already a baseline and track changes over the long term is definitely something that is very important to us, and we haven't really had the opportunity to do. And also, we know that what we do works in terms of changing people's environments, but we've been less successful at making a direct link to tracking whether or not it improves people's health. So we assume that if you put in a new food market, that people are going to make healthier choices, but it may not be the case. It may not be that if you build it, they will come. So having the opportunity to link these built and social environment kinds of changes to direct health outcomes, I think is a golden opportunity.
You do need to be careful about when you say things like putting a food market in a desert when you're in the Israeli environment. But no, it's an exciting prospect. Carlos, any thoughts about, in the extraordinary global universe in which your science functions? Any thoughts about how one can take advantage of this particular kind of opportunity?
Sure. So, as I've spent time thinking about the U.S. healthcare system and others, and how we can incentivize innovation, particularly around data sciences, what's turned out to be probably the two biggest pain points are data provenance as a just huge friction in the system that ends up leading to waste, right? So, when I sit with a guy like Dr. Newcomer, who heads oncology for United, and he says, look, I can't reimburse a gene panel test because the biggest problem I've got is I'm not sure why the panel test was ordered. I know why the CPT code is there, because that's the one that we reimburse the most, but I'm not sure why it was ordered. I don't know what genes are on those panels. I don't know why this test versus that test, and so it's very hard for me to make the right decisions, right? In closed systems like this, you can really begin to answer questions like, what is the proper deployment of genomics that improves outcomes and lowers cost? Right, similar to what Genome England is trying to do. And we've seen in this very room, right, 2 days ago, or last week when we had Snyder's conference, we had Dr. Nancy Cox from Vanderbilt come and talk to us about how they're using genetics to predict utilization. For example, they'll go through and say, what are signatures of utilization for cystic fibrosis patients, and then go back and ask how much are we actually utilizing, and realize, wait, we're utilizing 25% more than we have the number of diagnosed CF patients. Are we really missing? I'm predicting we have 400 CF patients, but we're utilizing like we have 500 CF patients. Do we really have 100 misdiagnosed CF patients? And sure enough, they had like 89. And like this, they went through, and every single Mendelian disorder, except for PKU, had a delta in the system, right? So they make a very, rational healthcare economics argument about why genotyping people as they're coming in the door eventually pays off, because you've got 5 to 10% of the population where knowing their genetics would be very beneficial to their management. Josh Knowles and FH, right? If we could identify patients who really have a genetic cause for their super high cholesterol, we should be tracking them so we avert heart disease, right? So I think in this case, we have a very unique opportunity to partner here, largely because of the size of the system, but as Manny is saying, because as we do the genetics, and we do the genetics to the extremes of the distribution, we may really find extraordinarily good entree points for drugs that could be prophylactic, right? PCSK9 in the FH setting is a prophylactic drug to avert heart disease, right? I mean, you could really keep people healthy and avert disease. So how many of those examples do we have out there? This is a unique place to study that. But I also think, as I think about the future of healthcare, it's all going to be about integrating the data. So going back to the data provenance piece, I think many of these technological hurdles are just going to fall away, right? People are building permission files in the sky using blockchain that in theory can allow you to link every piece of data about you to every other piece of data. And it's all about educating people and saying, okay, what permissions do I want to give to whom to bring this data together? So in that setting, right, we have a consumer electronics drive to have everyone walking around with an iWatch and monitoring devices. So we really invert the model. We begin to put together, huge reams of data with academics making the value proposition that, hey, guys, we should be here to help organize the studies so that we don't have this being disseminated by the companies that themselves are putting out these devices. Let's put this together in a real way to think about preventative and primary care as the main societal approach to averting disease in the future. And I think here, again, you've got a very good setup of how that's being deployed in a hospital-based model, where in the US we still have to figure out what is our model, because we're not deploying primary care in any, in a serious way. So that's the opportunity I see.
So let me take you up a notch and just remind you how
Take me up a notch.
potentially rich the opportunity really is. So first of all, so just to be clear, at the moment, although Clalit maintains a few biobanks, they're very small and selective. They do not have either material on their entire population, nor do they have it in silico, so they haven't genotyped their population yet. And with Manny, we're talking about the first focused sort of registry-based studies, picking a few diseases, probably inflammatory bowel disease being a fabulous place to start. But here's the setup. So this is a population, one of the most, I'm not hardly a computational geneticist, but I have come, as an Ashkenazi Jew, I have come to understand how blocked the genetics are of this population in terms of founder families. It's why there's such an incredibly rich, unfortunately rich rate of autosomal recessive disorders in the population. It's why I had to get tested before my daughters were born. And in fact, a huge fraction of the Clalit population, parents in the Clalit population, have been tested for Tay-Sachs and other rare diseases that aren't so rare in that population. The point being that they're not strangers to doing, at least both in newborns and in parents, in doing genetic testing. And I think it's an environment in which there would be considerable receptivity, even though they have some of the same craziness, privacy issues, and concerns that exist in other populations about the potential downsides of doing genetic testing. But here is a population in which they're doing things like Tay-Sachs the old-fashioned way. They're spending as much doing testing for a couple of rare variants as their rare alleles as they could for doing, completely genotyping the entire population. So the potential exists to actually do this kind of intervention.
And I would argue that the genetic data costs are trivial and are going to become ever more trivial relative to the total cost and value of the information. I mean, as everyone knows, diagnostics is a terrible business to get into because the margins quickly go away, and because it's just, you don't make money charging people for a blood glucose test, and you're not going to do that for DNA either. It's just going to be, absolutely necessary information, and I think it makes sense to really start deploying, similar to how the Brits are thinking about this, right? They've set aside money to sequence some, number of genomes a year with the argument that, if I'm not finding it with a panel test, I might as well just go and get a genome, because I've got a closed system and that information is going to be valuable to me down the road. And their pilots are proving that out. So, I don't think it's too early to think about that at all. And there are commercial operations out there that can do this at scale, where, we could really be on the data side helping drive the innovation, the learning healthcare system side, the healthcare economic side, the drug spin out side, and the genetics is, it's just like any other commodity that's out there. You farm it out.
Well, I think we need to talk about the possibility of doing this, which would, of course, make this one of the richest data sets in the world. So Jeremy, let's flip the discussion a little bit. So what can we learn? So one of the fabulous things about the Clalit and about the Israeli system in general is that there's very low levels of roiling. So one of the problems here is if you ever look at a claims data set or an MR data set, no one's in it for more than 15 minutes. I mean, except for Medicare, maybe the VA, in which people kind of settle in for life, so to speak, and their claims are there. But between commercial insurance, people come and go from their jobs, Medicaid, that people come and go frequently because of small changes in their environment or in government. We don't have in this country lots of longitudinal sets. Even a Kaiser, frankly, is subject to a 5-10% turnover every year. So if you really want to look at someone 20 years down the line, or you wanted to for purposes of looking at genetic association, look at grandparents, parents, and children from the same family, your chances are very small in most data sets. But here we really do have longitudinal data There's about a 1% turnover a year. It varies a little bit, but that's the range that it's in, so that very high fraction of these people, you've literally 20 years of data, and on a high fraction of those, you have either a child or a parent, and in many cases, both. So you've got, and they're demarcated in the data set, so you can actually find them. So what kind of things might we learn from such data?
Well, I think that Clalit in general poses a lot of different exciting scientific possibilities on these lines. It's something that, as from my neck of the world, it makes one salivate in some ways with the opportunity, and I can kind of explain that a bit by way of, if we think about something like a telescope or a microscope. If we imagine, you might not know very much about the history of either telescopes or microscopes, but if you were to think about it, you would think that the history would either be a late history, well, there hasn't been a lot of improvement in telescope technology, or that there has been a lot of improvement in telescope technology in recent years. The equivalent of a telescope or microscope that I am trained on using, and many social scientists are trained on using, is social survey data. And social survey data is, in fact, getting worse in a lot of cases, which is very unusual in the history of scientific instrumentation. It's getting worse just in the difficulties of getting people to do surveys, specifically, and also sort of issues of privacy around the information they're willing to provide in a survey. But while there have been exciting improvements in survey methodology, it's something where, in my existence, I almost feel bifurcated at this point, alternating between conferences about genomics or administrative data, where everyone is talking about the exciting opportunities, or things for my existence working with social surveys, where it's fretting and how can we sort of stem the decline of this instrument and save it for sort of as long as we can and renew it. And I think that makes these kinds of opportunities especially exciting, especially now that what Clalit was, in my understanding, is relatively early for electronic medical records as widespread adoption. So that means that the longitudinal data is over a longer time scale than we might otherwise have. Because of the system, as Mark said, one is able to connect spans of time, so in other words, within an individual, you have a great span of time because you have little, I guess roiling is the word, or little, to have little movement out of the system. And then because it's a system that enrolls half the people, you have a lot of families. And so in that respect, you've got this very nice structure. You've got siblings that you can put together, parents and children that you can put together over long spans of time. And I think that the opportunities there are very exciting. I think particularly if we think about, there are, in this world, I think that there's for any country, or any sort of data set like this, we could think about, well, what are the special cases of this country that it poses as scientific opportunities? So, for example, Israel and the United States, if we think about that comparison, Israel and the United States, by World Bank and other statistics, had extremely similar life expectancies from about 1970 to the late 1980s. Starting in the 1980s, that began to diverge. Israel has, in fact, done, it's a remarkably linear trend in the increase in life expectancy in Israel, whereas in the United States, life expectancy has increased, but at a slower rate. So now, I believe that the gap between Israel and the United States is over 3 years in life expectancy. That right there poses an interesting scientific puzzle about what it is about the two societies that has led to this change, that Israel has been able to maintain a linear increase in life expectancy, where countries in the United States and many countries in Europe have not.
So let me ask you this question, sort of a very, from my point of view, very complex, but I think probably practical question. When you think about the relationship between administrative data, by which I mean any data collected for purposes other than the research you're doing, so it would include, in this case, the claims and the electronic medical records. But one of the big differences between administrative data and survey data is that the survey data seems very purposeful. because it was collected on purpose for the research. Whereas EMR data and other forms of claims data and so forth, as administrative data, although they obviously contain, I mean, the advantage is that they were obtained for independent reasons, so that there's unlikely to be wild bias and things like that. But on the other hand, they're not necessarily very on target to what it is you might want to learn, especially about the way people live. So my colleagues and I are very good at writing crap about disease, but not always so good about writing crap about life. And if what you're trying to do is extract something about the way people live other than the odd terrible habit like smoking or drinking, you can't necessarily get that much out. In the Clalit situation, one perhaps enormous advantage, which is that, and unlike most other situations in which you get access to electronic medical records, we get, in this relationship, I should say they get the whole thing. Every single shard of a note, every little comment that a nurse or a health aide or anyone else made in relationship to care. Certainly the hospital records have all of that, things that you may consider wildly excessive. Putting aside the language issues, is there likely to be some advantage? I mean, do you think we may be able to learn something about people's real lives from their electronic records, not about just about their health, but actually about the way they're living their lives, their general attitude towards compliance, their health behaviors, things like that. And if it's not clear, is it worth doing an experiment just to see how, for example, the information EMRs might align with what you've learned about people in the Wisconsin longitudinal study or something like that?
Yeah. Well, no, that's an extremely interesting proposition. I mean, you could see both the great promise of being able, so basically taking what might seem incidental traces of people's lives that have been recorded, and can that be harnessed in a systematic way? Is there enough signal there that we can use it to interrogate questions? You could see that both sides. You could see the payoff if that works. You could see questions about whether or not it could either be so haphazard that it doesn't work, or it could be that there's some systematic bias. So for example, in the Israeli context, there has been there's some idea that diabetes mortality is higher in Israel than in other countries, but there's work suggesting that actually most of that is with differences in death certificate recording. So Israeli doctors being more likely to cite diabetes as an underlying cause of death, seemingly accurately than many other places. And so that would be the big issue, right? Is the signal that one finds there, are you actually uncovering signal about the person's life, or are you capturing signal in the recording system that's used for the life, or differences in that? And that is a very interesting scientific question. And so are there ways that we could get leverage of that? Because I agree that would be extremely useful if we could.
I mean, this is where the consumer electronics and medical devices comes in, right? Yeah. So if I think about, a company like Livongo that's trying to give everybody infinite strips and collect their blood glucose and hemoglobin A1C levels, I marry it with what you're talking about. Well, now you got data, right? You can track this, and you've got the information, pre-autopsy, whatever. You can, really, begin to see whether that's really happening and compare it to the United States and other places. So I don't think, I think bringing data together in the future is just not going to be an issue from a technological point of view. You're going to be able to bring together medical record, military records, if you can get access to them, school records. There's not going to be a limit. It's going to be a question of what are we comfortable with, what are people going to give consent to, and my feeling is that this could be, for us, for a few years, a wonderful place to begin to sort out what that looks like, and perhaps bring in the traditional survey methods and deploy them in a very controlled setting where we've got orthogonal pieces of data that we can bring in. To me, it makes a tremendous amount of sense.
Sandy?
And I would just like to tap into the other part of the research that I do is a lot of what we've been talking about, when we talk about somebody's health, we're not actually talking about their health, we're talking about their illness. And I know that Manny was referencing in some of his slides using some of the genetic information to look at things that can protect people against certain diseases. So I would say that not only can we use the genetic components, but we could use these kinds of longitudinal data that have a holistic perspective about people that take into account their education and where they lived and their family situations, to look less at illnesses, but more about the circumstances under which people thrive and obtain their full potential. And I'm sure that all of us know people, some people, their plates get really full with a small amount of disasters, and other people just seem to absorb the most terrible things, and they just keep on, you know, moving along forward. So what is it about people that improves their sense of well-being as well as focusing on illnesses?
Yeah, that's a great point, because when I've talked to our colleagues and about what the U.S. spends on healthcare versus others, they say, well, also realize that it's not just healthcare, it's healthcare plus social services, and basically everybody spends about 30%. We choose to spend 20% on the healthcare and 10% on the social
nothing
services.
on social services.
Right, and they spend the other way around, but they're actually kind of covering a lot of the same services sometimes, right? You know, where are social workers deployed in the United States? You know, more in the medical system versus in, you know, in community, where the other places do it logically.
Right, and their life expectancy is rising and ours is flat.
So, but I mean, and again, but this is a place where that, again, it's a closed system, so you can account for that, right? It's hard to account for those externalities in analysis of U.S. because, you know, there's just so many things going on simultaneously.
Yeah, no, I agree.
You know.
Sandy, just to go back to you a little bit, so in the early work that you've done that's beginning to scale up, are there particular social or environmental eccentricities or aspects that merit approach or remedy, but which would also merit a investigation? I mean, assuming that the 50% of the population that you could geocode right to the areas where you've identified an environmental issue or a social issue could be linked, and assuming that the population is relatively unbiased in terms of who's in Clalit as opposed to who's in one of the other three healthcare systems, to which incidentally there's also access to the data, so you could even challenge that hypothesis if you wanted. But are there particular things that have been learned already from the discovery tool that might be the nidus for a health check, which is to say to see whether or not there is an association between those conditions and something observed either in quality of life or in in illness directly as you've used it? That would be potentially actionable? I mean, are there some things we could do just today from things we've observed?
Well, I think that the thing that surprised us about doing this work internationally is how common some of the issues are across different countries and different cultures. So that speaks to a sort of a very holistic approach in uniting communities around these built environment issues. I don't think that there was anything specific that, I mean, any built environment changes that you make are going to serve the population. So you can imagine if you make sidewalks safer for older adults, you also make them safer for the woman who's in the stroller or the person who's disabled. So I would say it's not so much the differences, but the commonalities that can unite people and provide a better way of moving forward with solutions.
Great, thank you. So Manny, I don't want you to feel left out. There was one very sort of family-oriented question that I wanted just to make sure we cover a little bit, which is again one of the most striking things about the Clalit environment is the fact that families tend to join Clalit and stay in Clalit over generations. So we have the generational thing. And I gather even without genotyping the entire population, and even before we do the huge experiment that Carlos has been proposing for us to do, that we could learn something about heritability in these populations. And I wonder whether you just want to comment on that.
Yeah, that's correct. If you have phenotype information or medical information about grandparents, parents, and children, analogous to what was done by Fisher and Mendel early in the 20th century, you can start estimating genetic parameters like heritability or how components of disease risk are shared across multiple diseases. So I think it's a phenomenal opportunity to use the existing data without genetics to actually learn about genetics of diseases and health measurements.
Right, and in order to do this, how intrusive or invasive or expensive do we need to get?
So it would mainly be about inferring familial relationships across individuals in the healthcare system. And I think that information is available right now. And the second thing would be, or the second task would be to integrate that with the medical and health information that's also available through Clalit. So not intrusive at this moment.
So this is an in silico challenge that would require no new kind of informed consent for blood for new tests. This is just taking advantage of expressed phenotypes and expressed or, basically professed relationships among.
That's correct. And the other opportunity that exists is using the longitudinal data to learn about how heritable some of the disease progression components of disease onset is. And that is something that we don't know much about at this moment in time.
Great. Jeremy?
Well, so this actually raises an interesting question that I hadn't thought about. So in Clalit, you would then be able to identify twins in Clalit, right?
You bet.
Yeah, and so normally like if you're doing research with twins, you have one of two routes. You can impanel the twins in a twin study or something like that. That has obvious sort of representation issues and issues in terms of the data that you get. The work that's been done with administrative data, for example, I've done some work using matched birth and school records from the state of Florida with some folks. You can't identify, there's not information about the zygosity of same-sex twins, right? And so you don't have leverage. You have to use this indirect form of leverage. But
right. I bet you do with same-sex twins. I suspect that there's a notation as to whether they're viewed as identical or not.
Yeah, and they would overwhelmingly be in the same system. So we might talk about that later.
Yeah, many opportunities. So we have a couple more minutes. We'd be more than happy to take a question or two from the audience if there are. We are recording the events of the day, and for this session and for the 4 that will follow it, we encourage people to come to a microphone. Rita has her hand up over there. You can see one microphone. Leslie has her hand up over there. There's a second microphone. We have 5 or 7 minutes, so feel free.
Daniel? Thank you all for your excellent presentations. I have a question on your studies on inflammatory bowel disease. Have you looked at associations with the development of cancer, which is, as you know, very high in such patients?
That's a great question. As you may know, most of the genetic studies are limited to a particular disease. I think one of the great opportunities that exists with these large population scale studies is the ability to look across diseases. And that would be a phenomenal question to pursue in Clalit. So the answer is no, but there's a great opportunity.
Particularly now that we're seeing in this country earlier onset in patients 40 to 50 years of age rather than the older age group getting an increased colorectal cancer.
Again, this is a perfect example of why the collection of the genetic data is trivial in comparison to the rest of the system, right? Once Manny has the variants that he's associated with inflammatory bowel disease, it's very easy for us to go into our own system and ask, do patients that carry those genotypes have an increased risk of cancer, right? Are they overutilizing, cancer resources relative to controls, right? And those are, now routine FIWA studies that in fact, began as a way to understand the genetics of the system and pleiotropy, right? How many times does one mutation have a bunch of effects? And is now becoming a standard tool of economics, right? How do I use this information to predict what people are going to utilize so I can avert that problem in the future?
Great. Any final comments before we give people a little stand-up break? Anyway, I want to thank the panel. This has been good.